Hello everyone. Let's start with the next biomolecule, that is nucleic acids. In this video lecture, we will go through some of the basics of nucleic acids. We will understand regarding its basic unit, which is a nucleotide. So, nucleic acids are large biomolecules. They belong to macromolecules category. These are involved in storage and expression of genetic information. Right? So the two types of nucleic acids, that is DNA and RNA, these are involved in storage of genetic information. They act as genetic material, and RNA also helps in expression of the genetic characters. DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. The basic unit of all nucleic acids is a nucleotide. So whether we take DNA or RNA, the basic fundamental unit of any nucleic acid will be nucleotide. Let's understand what's a nucleotide. So nucleotide is composed of three parts. First, we have the pentose sugar. Pentose means it will have 5 carbons, right? Pentose sugar is the first part. Next, we have nitrogenous base. Next, we have the phosphoric acid. Simply, we call it phosphate group, right? So, every nucleotide will have these three parts: pentose sugar, nitrogenous base, and phosphoric acid. Let's understand which pentose sugars are found in nucleotides. So, ribose and deoxyribose are the two types of sugars. These are monosaccharides, simple sugars, which are found in nucleic acids. Ribose, see the structure, right? It has 5 carbons. You can see the position also. This is carbon number 1, this is 2, this is 3, this is 4, and this outside is the fifth one, right? Also, if you compare this structure over here, deoxyribose, there is only one difference between both the structures. Rest part is the same, copy paste. On the second carbon, what can we see here? Here we have OH in case of ribose, and in case of deoxyribose we have just the hydrogen. This is the small difference which is present between both the sugars. These are the pentose sugars, 5 carbon sugars. Ribose sugar, this one with the OH group, right, on the second position. Ribose sugar is found in the nucleotides of RNA. That is why it's called ribonucleic acid. It has ribose sugar. And DNA nucleotides have this sugar over here, deoxyribose, right? So all the nucleotides in any DNA molecule will have deoxyribose sugar. That is why it's called deoxyribonucleic acid. So the first half of the names, ribonucleic acid and deoxyribonucleic acid, actually it comes from the sugar which is present in the structure. So ribose in case of RNA and deoxyribose in case of DNA. The next part is nitrogenous base. Now, nitrogenous bases are of two types, purines and pyrimidines. Purines, we have two examples over here, adenine and guanine, right? Adenine and guanine. In short, we just write them as AG. Purines, okay? Done. Next, we have pyrimidines. So, we have cytosine, thymine and uracil, right? These are written as C, T and U in short. Purines are double ring structures and pyrimidines are single ring structures. You can see the structures for your reference. These two over here, double ring structures, are purines, adenine and guanine. And these three are the pyrimidines, cytosine, thymine and uracil. These are the 5 types of nitrogenous bases which are found in nucleic acids. Now, out of these 5 types, adenine, guanine and cytosine are present both in DNA as well as RNA. Thymine is present only in DNA and uracil is present only in RNA, right? So in short, we can say that adenine, guanine, cytosine and thymine are present in DNA. Then adenine, guanine, cytosine and uracil are present in RNA. The third part is phosphoric acid or the phosphate group. So this is simply the H3PO4 molecule, right? This is also present in every nucleotide. Now let's see how to join these three units: sugar, right, the pentose sugar, nitrogenous base and phosphate group. How to join these three and form a nucleotide. So we have taken the sugar. So here I have drawn a basic representation. This can be ribose or deoxyribose, right? So we have taken the pentose sugar. Now to the first carbon here, first position, we attach nitrogenous base. Depending on the sugar, depending on the kind of nucleic acid, we will have specific nitrogenous base. Like if we have, let's say we have taken ribose sugar, so it should be nucleotide of RNA, right? Then nitrogenous bases which are possible here are adenine, guanine, cytosine and uracil, right? And in case of DNA, we will have thymine. So nitrogenous base is connected to the first position over here. Next, to the fifth position here, fifth carbon, we attach the phosphoric acid, the phosphate group. So we have connected all the three groups: sugar, nitrogenous base and phosphate group. This is a nucleotide. It is also important to note the bonds which connect these three structures. So nitrogenous base is connected to sugar with the help of N-glycosidic bond, and phosphate group is connected to the sugar with ester or the phosphoester bond. So these two bonds, N-glycosidic bond and the phosphoester bond, helps to connect the three structures to each other and form nucleotide. There is one more term which we should understand, that is what is a nucleoside. Okay. Now all these three things put together, phosphate, sugar and nitrogenous base, this is a nucleotide. Now we have to understand what is a nucleoside. This part in the box, sugar plus nitrogenous base, right? This is nucleoside. So can we say that nucleoside plus phosphate is nucleotide, right? It is also important to note what are the various terms we use for the various nucleosides and nucleotides. Okay, so let's go through it. Here I have made a very easy table for you to visualize. Here in the first column we have the nitrogenous bases, and here we have specified whether the nitrogenous base is present in RNA or DNA, right? That means to which sugar is it attached, ribose or deoxyribose. In the next column we have mentioned the names which we use for nucleoside, and in the third column we have mentioned the terms which we use for nucleotide. So simple. Let's start with adenine. Adenine is present in both RNA as well as DNA. That means adenine can be connected to ribose as well as deoxyribose. So if we take ribose, sugar, and attach adenine nitrogenous base, this will be a nucleoside. This is called as adenosine. And if we attach a phosphate to it, it will become a nucleotide and it's called adenylic acid or simply adenylate. If we talk about DNA, right? Adenine is also found in DNA. So just take deoxyribose sugar, attach adenine, we will get the nucleoside deoxyadenosine. And if we attach the phosphate, we will get the nucleotide which is deoxyadenylic acid. The second one, we have guanine. This is also present in both RNA as well as DNA. So take ribose sugar, attach guanine, you will get guanosine. Attach phosphate, you will get the nucleotide guanylic acid. Simply guanylic. Next, in case of DNA, we will take the deoxyribose sugar, attach guanine, we will get deoxyguanosine. So just add a prefix deoxy and just copy this term. We will get the nucleoside which is found in DNA. So here deoxyguanosine, and the nucleotide is deoxyguanylic acid, right? If we take cytosine, then again take ribose sugar, attach cytosine, you will get cytidine. If you attach phosphate, you will get cytidylate acid which is cytidylate. In case of DNA, take deoxyribose sugar, attach the cytosine, you get deoxycytidine. And if you attach phosphate, you get the nucleotide which is deoxycytidylate acid or deoxycytidylate. Next, we have uracil. It is present only in RNA. So, we will just take ribose sugar, attach uracil, we will get uridine. And if we attach the phosphate group, we will get uridylic acid, simply uridylate. And last, we have thymine. It is present only in DNA, right? If we take the deoxyribose sugar and attach thymine to it, we will get deoxythymidine. And if we attach a phosphate to it, we will get the nucleotide, which is deoxythymidyllic acid. So here are all the terms which we use for nitrogenous bases, nucleosides and nucleotides. First, memorize all the names which we use for nitrogenous bases: adenine, guanine, cytosine, uracil and thymine, right? Next, if you look at all the names for nucleotides, right? Adenylic acid, guanylic acid, cytidyllic acid, uridylic acid, or simply adenylate, guanylate, cytidylate. So either it will end with acid or like late. So it is very easy to remember in this way. And the remaining terms are definitely the nucleoside. So adenosine, and if there is deoxyribose sugar, then deoxyadenosine. So you can simply remember all the terms mentioned here, and in case of DNA, just attach deoxy. You get your answer, right? Uracil will be only in RNA and thymine will be only in DNA, right? So these are all the nitrogenous bases, nucleosides and nucleotides. So in this lecture, we have seen the basics of nucleic acids, what is a nucleotide, what is a nucleoside, and the terms which we use for all these nucleosides and nucleotides. So now it's time for knowledge drill. So try to tell the answers for these questions in the comment section. Name the nitrogenous base found in RNA only. And second question for you is identify the nucleosides from the following. So I am giving you some terms: guanosine, uridine, cytosine, adenylate and thymine. Out of these five, tell me which are nucleosides and tell me in the comment section. I hope this video was helpful for you. Thank you.